And so now I'm going to pivot to introducing Matt. We're really pleased to have Matt Boucher here. He's from Penn State. Matt has received his BS in biology and anthropology, go anthropology, from UMass Amherst, and went on to a PhD at Cornell, working with Greg Loeb there. And there he worked on his manipulation of insect vector behavior by a bacterial pathogen of apple, and completed that in 2020. Since then, he's been on the PSA project at Penn State, working with John Tooker's lab, managing the insect end of CE2. So we're really happy to have you, Matt. Go ahead and take it away.
All right. Yeah, so thanks for that introduction. Thank you all for being here. I'm pretty excited to share this data with you guys today. There is a lot. I did a little back of the napkin calculation, and there is potentially up to 300 different ways I can analyze these data. So part of the goal for this this project is to not only kind of share with you what we found so far, which is really exciting. There's some really nice crisp takeaways from these data, but also to think about and discuss different ways we can look at them. And as we roll into year 2 of this project, what potential things we could look out for as different ways to add to these data as we go. So I'll kind of, just as a layout, I'll go through a little bit of an introduction. We probably don't need much here besides more in the insect realm of things, so I'll focus on that. We'll go into the basic methodologies of what we've done, but I've also presented that numerous times here at the team meeting, so I won't spend too much time on those. And then we'll just dive right into these data, and then hopefully open it up for discussion right at the end for a little bit of time to discuss different ways to look at it. So you can see here's our beautiful field site in Pennsylvania. We're planting into the rye, and you've got your planting green treatment, your planting brown treatment, and your planting green-brown treatment, and we've got our control no rye up here. You can see it out there in the back too. So we're hoping for more of these beautiful days as we move on. So I think the important thing is to start with no-till. So no-till, obviously, in the Mid-Atlantic is a big deal. Across the country, it's kind of growing. So the darker shaded colors here is where proportion of croplands in no-till based on the 2017 USDA data. It's expanding, and that's great for all of the conservation benefits that we already know here in this group today. But it also means that there are new problems. You can't just have a solution without having more different issues, more and different issues, I should say. And so early season pest issues can be pretty significant, especially in places like the Mid-Atlantic, but we can also see them across the country. You get sporadic pests such as true armyworm, whose damage is kind of what this looks like down here. They kind of shred the sides. They don't like the midribs. You also get black cutworm. So black cutworm will either trim the plant right at its base and pull it into a little burrow and consume it, or it will make these kind of little hole punch style damage markers there. In the Mid-Atlantic and increasingly in the Midwest, where a lot of our field crops are produced, excuse me, sorry about that, slugs are a major issue. So this is a field, as stated, that was replanted 3 times due to slugs. Slugs like it wet. Without tilling the soil, basically, the eggs and immatures and adults don't get destroyed, so they just persist, and a single slug can lay up to 300 eggs. So it can get pretty bad pretty quick. This is one of John Tooker's kind of quintessential slugs are a problem photos. All of that kind of debris you see there is just slugs. That's fistfuls and fistfuls of slugs. And that's just a standard hay mower there. And our main slug problem in the Mid-Atlantic and growing in the Midwest is this gray garden slug here. The gray garden slug was an introduced species from Europe. It was introduced in the 1800s, and it's been problematic ever since. Again, slugs like wet weather. One of the problems with managing slugs is that their pesticides come in bait formulations, but those baits tend to dissolve when it's wet. And the problem is slugs are a wet weather pest. So the management tactic doesn't really line up. And in conjunction with that, it gets very expensive to continuously apply these baits if you have a slug problem. So here's just a couple statistics here, some slug eggs here. They're very beautiful kind of, when you see them, they have a really opulent color structure and they reflect light very nicely. It's quite pretty, but they are quite problematic. So here's some kind of, things that I have gone over already. And down here, you can see what the damage looks like. So oftentimes slugs will leave a slime trail. That's a pretty good indication that that's what's there, that's what's causing your damage. And they leave these kind of strips because they kind of rasp along the plant. So you'll see that a lot when you see slug damage. And here's a little bit of a slime trail right here from this little slug that I took a picture of recently. Our big question is, can cover crops help protect no-till fields from invertebrate pests? We know that we can use cover crops for weed management. We know the conservation benefits of cover crops, but there's also this potential to expand what they can do by showing that they can protect against invertebrate pests. Cover crops, as we know, has been increasing in usage, especially in the mid-Atlantic in the last 10 or so years. And the most popular is cereal rye, which is what we use in our experiments here. Cover crops have multiple mechanisms through which they can potentially help manage invertebrate pests. One is by creating habitat and alternative prey inside that residue. So that residue has a nice microhabitat. The predators like carabid beetles can live inside of those plants or live inside that residue. There's also a lot of alternative prey that persists there longer. So soil microarthropods, earthworms, pretty much anything that a predator eats, there's more of in that residue. And so that's really great because it can keep those predator populations high and it can keep them present rather than in a no cover crop situation where the predators might just be kind of transient or migrating through as they search for a better place to find food. There is some data out there that slug damage incidence decreases as predator populations increase in cover crop fields. So this is a figure from a project that John did a while back, and you can see on your y-axis, that's the slug damage, the proportion of plants damaged, and the early season predators. So a correlation here between as number of predators increases, proportion of damaged plants decreases. So we have some resolution on this top-down control idea, but we need a little bit more. There's also planting green, which obviously is our main focus of CE2. And we think that this can make the pest suppressive benefits of cover crops even better. That being because that residue is going to stay longer into the field as it decays. It might create a better habitat for those predators. And it also might create a dietary distraction. And so this is the bottom-up control aspect of this. So essentially a two-pronged approach to managing these pests, where that decaying cover crop has a lot of, again, alternative prey and et cetera, kind of living in the residue. For slugs especially, and for other pests, slugs are more consistent, whereas kind of black cutworm and true armyworm are more sporadic. So this is kind of why the focus is on slugs here. Slugs are essentially omnivores. They will eat anything. In the field, I've observed them eating dead mice. I've observed them eating just like fungi, right? So they'll eat pretty much anything, and you get a lot of that stuff going on, and they'll also eat decaying plant matter. So the idea here is that it can pull those slugs away from the crop into the residue and keep them contained there. And we actually have data to show this. And so I recently co-authored a paper that just got published 2 days ago. It's out. I will send it through the PSA listserv to kind of show what we've done. But the basic takeaway that we had here was that there was a lower damage probability in planting green plots despite equal top-down control. So this figure here is the probability of predation in these in planting green fields. And you can see with our bare treatment, our planting brown, and our planting brown being our kind of standard cover crop management, and our planting green, there is no difference in predation rates there. But what you do see is a lower damage probability. And even more so, to bring your attention, we did this, we split this assessment between seeds that were coated with an insecticide and naked seed. The reason being that coated seeds can also have negative detrimental effects for predators. They can be toxic to predators, and slugs that feed on coated plants can accrue toxin in their bodies that then when predators feed on, they can receive a toxic dose. So what you see here is, and so these indicators here mean that this is statistically lower than all of these. So our planting green treatments have a lower damage probability, and they're roughly equal between coated and uncoated seeds, which is a potential area to save farmers money by reducing the use of coated seeds, and was kind of the impetus for us choosing to not use naked seed in our CE2 experiment, which we'll get to in a few minutes here. So that kind of brings us to our big question here, which is how does planting green fare as a pest management strategy across the United States? We have some data from Pennsylvania. We need to expand this. That's what PSA is here to do. And so that's what I'm going to talk to you about. Let's see. All right, so the project layout is pretty straightforward. You might have seen this numerous times before, so I won't spend too much time on it. We've got 16 states. Each state has a setup of we're all planting rye, we're all planting corn, and then we have 3 or 4 treatments depending on the state. The check treatment, no rye, the kind of standard management, which is 2 to 4 week termination, then planting green 7 to 10 days after, and we might be shifting this based on some feedback from farmers to a little bit earlier, closer to planting, and then an optional shortly before, which we're calling our planting green brown. So here's our project layout. Again, kind of standard field design. And then we've got different subplots for the different disciplines involved in CE2. And here's, so we kind of operated in the middle sections of these plants, or of these plots here. And so on your bottom left, these are all the different types of data we collected for this project. And because of time, and just because there's so many different ways to look at it, we're only looking at a handful of those today. So these are the underlined ones. We're going to touch on some of these. We're going to go in depth on others, and it's going to be great. So method, slug activity density. So slugs are a big problem in Mid-Atlantic. I'm going to keep saying over and over, this can be and probably will be an increasing problem in the Midwest. So it's really important that we get an idea of what's going on with slugs as they start to expand in terms of being problematic. It's really easy to sample for slugs. You give them something that keeps the ground moist underneath it, and they go underneath it. They like to be moist. They don't like the dry. They will find something to hide under. So we have little 1 foot square white roofing shingles, and there's 4 per plot, and those are placed in between rows 6 and 7 and 10 and 11. And then we count the number of slugs from planting to roughly V6, and then again from R5 to harvest. This gives us an indicator of what's going on in the spring, in the early season when they're problematic for the current season. And then that fall data gives us an idea of what the slug pressure the following year will look like. So that fall data is really important because it can give us an idea if those planting green plots might create a compounding effect for slugs that might make it untenable to use multiple years in a row or something like that. Early season herbivory here, in each plot, 5 transects, you count the number of plants, that's your stand establishment. You count the number of plants damaged by invertebrates, that's your damage incident. And then you categorize the damage, and we only chose a few here. So slugs, black cutworm, true armyworm as the damage types we were interested in 2021. And 2022, we're going to add stink bugs to that because there was a fair amount of stink bug damage across the board. And then you also rate the damage on an ordinal scale. We will not talk about the damage severity today. That's a huge data set with over 50,000 rows of data. So I'm still really just digging into that to start. Our predation rate is done by these sentinel prey assays. So the foundation of that is you get a little waxworm, you pin it to a clay ball, you bring it outside, you put it in a cage to make sure invertebrates can't get in. A little lid goes on top of this cage and to protect from anything coming in like a bird. And then you check them. And so for this project, we check them twice. We deploy them at 8 p.m. one day, then 12 hours later, we check them. That gives us an indication of nighttime predation. And then 12 hours after we check for nighttime predation, we check again, and that gives us a measure of daytime predation. Predators mostly come out at night, so we're going to see higher daytime or nighttime predation, but across the board, we get an idea of total predation activity in the field. And you can see here, we've got a carabid beetle feeding on one of our waxworms, and you can get these images pretty consistently, but this was a pretty fun one. I thought it was nice to show that this actually works. I think when I started this project, I was like, this doesn't work. This doesn't make any sense. Why would this happen? But it does. And so that's pretty interesting. We're going to jump right into the data now. So we'll start with spring slug activity density. Excuse me. And so spring slug activity density varied by state. That should be pretty consistent. Some states didn't participate in the slug activity density measurements. Texas, Florida, Kansas, states like that don't really have slugs yet. We don't know if they will ever have some sort of slug pressure at any point. I think that the easiest way to kind of show you significant differences here is after giving you a second to kind of look this over. So you have mean slug activity density as your y-axis. On your x-axis, you've got your treatments. And you can kind of see in your planting green treatments in Pennsylvania, we had higher slug activity density. But then you look at a state like Virginia, and they had lower slug activity in their planting green plots. So some confusing things here, some that were more consistent than others. And again, in Maryland, you had lower activity density in planting green. The easiest way to kind of show you some significant differences here, I felt, was to just lay it out in a table. So these red arrows mean you have significantly higher slug activity density relative to a control. So this is relative to no cover crop use at all. And so you can see our overall model shows generally each one of these treatments individually has a higher overall slug activity density than our control. You can see in PA, we have higher in green. You can see in Delaware, Virginia, though, you've got lower, and this is relative to a control. The other important thing is that we're not always comparing this simply to the no cover crop option. So this table shows you differences in spring slug and slug activity density relative to that planting ground treatment or standard cover crop management. And what you see here is not as many differences. So if you're using cover crops, right, planting green doesn't necessarily increase your slug activity density. And I think that's a really important takeaway for this talk, is that if you're using cover crops, you might not be seeing such a significant increase in slug pressure. That's like something we commonly hear, is that using cover crops, planting green is going to increase the amount of slug activity in your field. And what we're seeing is that delaying the termination time, if you've already committed to using cover crops, might not be as problematic as one would imagine. So now we'll jump into the fall, and the fall data is really interesting, and this is where the story of slugs gets really cool. Let's take PA. We'll go back a few just to give you a second to look at this. In PA, the planting green slug activity density in the spring was the highest of the treatments. It was significantly higher than the control. It was not significantly higher than the standard cover crop management. But in the fall, this is reversed. So the slug activity density in the fall in Pennsylvania is not as high. And not only is it not as high, it's significantly lower than your control here. And so that's what this arrow indicates. And so what this data across the board tells you for the fall is that the differences in slug activity density between the treatments, including the control, essentially dissipate over time. And that's really important because that means that, again, using cover crops and using planting green might not lead to increased slug pressure over time. And that's really important for getting these practices in continual use. So I find that to be incredibly interesting. Again, here I'll just go to this to show you the significant differences. There essentially were none, except with the exception of Pennsylvania. There were no differences. And this could be driven by a number of things. This could be driven by predation, right? To get some resolution on this, we'd have to do a little bit of a different experimental setup because you can have, one of the potential things that's happening here is that predators are building up in population in the planting green plots and then migrating out and feeding on everything in those plots. And so those plots are kind of close together, but beetles can move as far as they can move hundreds of meters in a day. So if they're out foraging, they might find something in the control treatments. So to get some more resolution on that, we'd have to set up a different kind of experiment. But in general, this is really productive and important data. It shows that slugs are not as problematic, and that the cover crop treatments that we're trying to apply here might not create consistent problems with slugs. So in general, we'll move to the stand establishment data now, because one of the questions with planting green is that generally there's a thought that there's going to be a yield hit. Some of anecdotally with discussions from some of our farmers in Pennsylvania who have been using planting green consistently, they have now gone from having an initial yield hit to seeing yield benefits. That's anecdotal data. I don't have it to show here, but it's important to show stand establishment because that's an important metric of what yield may look like down the road. So here in our overall model, down here on the left, you can see there is no overall differences, but again, those differences do vary by state. And so this is at V3. And so the colors here in this table indicate significant differences from the control, significant differences from brown, and then significant differences from both control and brown. So if you take PA down here, it has a significantly lower stand establishment than both the control and the brown. However, there are states where most states that's really not the case. And in some states, so North Carolina, which is a very interesting story that we'll dive into here, actually had significantly higher stand establishment in their cover cropped fields. North Carolina had a major outbreak of cinch bug this year in 2021. And what we're seeing here is that potentially those cover crops actually conferred some control and they might have served as a dietary distraction, or they might have had higher predator numbers in those fields that helped control the outbreak relative to the control. So that's a really important piece of data in North Carolina. And so North Carolina and I had spoken a lot because they were like, basically, we couldn't get any data out of because the outbreak was so bad. But here again, even with the outbreak being very severe, we're seeing some important resolution on what cover crops can do in the face of a pest outbreak. And that's the really cool thing that this project is going to allow us to do, is over the next 3 years, someone else is going to have a different pest outbreak, and we're going to see how these different cover crop treatments support the cash crop through that. So you can again kind of see this resolution here in Wisconsin, where they had a lower stand establishment here. Virginia had higher stand establishment, which was generally against the trend at V3, but it happened in a few states. Kentucky's another one here. Similar deal at V5. So we did this assessment twice at V3 and V5. And so here's our stand establishment again. Things were a little bit more consistent. But in Iowa, Illinois, and Pennsylvania, we did see significantly lower stand establishment. Now, I share the stand establishment data with you for the agronomic aspects, but also in support of our damage incidence data, which is quite interesting. So damage incidence is the proportion of plants damaged by any pest, so any insect pest. Or any insect or slug pest in our assessment here. So you have your damage probability on the Y, and then you have your treatments on the X here, each state across the top. And so what you can see is that this tended to vary by state. You had higher damage incidence in Pennsylvania in cover crop treatments relative to control. So if you go down here, you've got your planting green in Pennsylvania, and it is significantly higher damage incidence than your control, but not significantly higher damage incidence to your brown. So again, when you're using standard cover crop management, you are also, and you're thinking about planting green, you might not see higher damage incidences as you would expect. You might see them higher relative to no cover crops, but in a lot of instances, at least in Pennsylvania, that's not really the comparison that's being made. So this damage varied by state. Again, so Kentucky was a really interesting one where they had significantly lower damage incidence in their planting green plots. And so a couple states showed lower damage incidence in their planting green plots. That's great to see. One of the things we can think about is why, because that's not really a consistent data point in this data set. And again, we can dive into this 1,000 different ways. The differences in damage incidence at V5 are less pronounced. So your plants are a little bit older now. You can see in different states, right? So relative to the control, damage incidence isn't as significant here, which is kind of consistent with the fact that stand establishment was more consistent at V5 too. And so your overall model, again, you're seeing higher damage incidence than both the control and the brown, but at the same time, when you break it down by state, those differences are they continue to be variable. And so again, Ohio here has lower incidence in its planting green plots, but Pennsylvania still has higher. So just, things are going to vary by state, and breaking that down and understanding that is something that I want to kind of open up for discussion towards the end of this assessment or this talk. All right, we're making good time here. So to kind of go back to our slug activity density, we're going to look at the different types of damage that the damage. So we just talked about damage incidence. Now we're going to talk about the different types of damage that we saw in those plots. The main takeaway here is that higher slug activity density in planting green plots did not translate to a higher proportion of damaged plants being attacked by slugs. So that's really interesting. Basically, there are more slugs in these planting green plots, but they are not attacking the cash crop at a higher rate. So what you see here in this figure is damage probability on your Y. Your X is your treatments again. And then you've got slugs, you've got black cutworm, you've got true armyworm, then you've got other, so different pests that were attacked, and then you've got plants that were attacked by multiple pests. So as you can see across the board and the overall models, there was not a lot of other pests involved. These were, again, sporadic and varied by state. I haven't gotten down to that resolution of state yet. I thought that this was a really interesting data point, and I wanted to make it, to bring it up and kind of show the initial findings here. So the focus here is on slugs, black cutworm, and true armyworm, really. And so what you see is significantly higher likelihood of plants being attacked by black cutworm in planting green plots, but not by slugs. And so that's really interesting. So those slugs might be living in that residue. Maybe it's better quality for them to survive, but is not actually translating to more damage. And so that's really cool resolution on this project that we haven't really been able to get before. This is kind of the first time that we're really going to be able to share, like, yes, you might see higher slugs, but you're also not really potentially going to see higher damage. Those two things are different. And getting the resolution on that is going to be a really important data point as we try to expand the use of planting green here. Same thing at V5, and it's actually even more interesting when it comes to slugs, because now you're seeing at V5 a higher rate of attack by slugs in the control plots. And so here you are, you're planting greens down here, you're planting, your control is up here. Again, to point out, in general for slugs, the damage probability is actually pretty low here. It's over 25% it's at 30% for the control, and it's down to about 10% for planting green. And so, you know, as I dig down into the individual states, I might get more resolution on why this is happening. But that, again, just really underscoring that when you have more, you might have more slugs, but it might not translate to higher damage. Black cutworm continued to be our highest damage incidence pest. So that's interesting in the with respect to that seemed to be across the board. Everyone's major problem was black cutworm, which is considered a sporadic pest, and true armyworm is considered very sporadic in general, at least in PA. So these data are kind of consistent within Pennsylvania and elsewhere. But again, we don't know, right? This year it might be that we get multiple true armyworm outbreaks during the early season, right? And so that's kind of what these 3 years of data is going to give us resolution on, is when you get a pest outbreak, how does planting green respond? So we'll have this base level data, but we'll also be able to say in North Carolina, when they got this outbreak, cover crops protected them, right, relative to a control. And so that's, again, more important in new data, because there's a lot that's not known about how cover crops, even outside of planting green, benefit pest suppression. And so that's really key to these data. Now we're going to dive into predation. And this is where the story, just like sometimes you just get really good data and you're like, I should quit right now. And this is what these data look like for the predation. Just very beautiful, perfect, nice data. And I don't think this has ever happened to me. And so I'm really excited about this. I was analyzing these data last night. So we looked at predation at V3, V5, and R3. We looked at it at R3 to see if there's a legacy effect. If that predation is high at V3, is it still high at R3? And so predation was higher in planting green at V3. And we can get that resolution for nighttime sampling, daytime sampling, and overall predation. So predation at V3 was in planting green was about 85%. 85% of our sentinel prey waxworms had been predated, relative to a 75% and a 65%. That's pretty awesome. That shows that planting green is keeping those predators there, or that's where they are, right? And you're seeing it not only resolve overall, but also at night and during the day. And even during the day, that's more pronounced. And that's really interesting, because most predators are active at night. But when they're active during the day, right, it makes sense that they would be able to predate more or forage more in those planting green plots, because that habitat's better for them. There's more going on there. There's a better microclimate, et cetera. So this daytime data is really interesting, because what it might indicate is that we're increasing the amount of time that predators become an effective biological control in these plots. And so that's really cool there. Same thing at V5. So what you're also going to notice is just a general trend of increasing predation rate. Of increasing predation rate. This is the wrong table here. Yes, that's, I put the wrong table in. So ignore these data down here. I apologize for that. But what you're going to see is a trend of increasing predation, because beetle populations build over time. We know that that's pretty consistent across studies. So you're going to see higher rates as you get later into the season, but you're still seeing this resolution here, where planting green, your predation rates are still generally higher than your standard management options. And then R3, oh, I just had two in there. Excuse me. Apologies for that. Then we'll touch on R3 here. Again, overall, you're seeing almost 100% of your sentinel waxworms being predated. Most of them are being consumed at night. The remaining that are consumed, again, are still, you're getting higher resolution during the day in terms of predation in planting green and planting brown than your control. That R3 data is a little bit more difficult to analyze, because you're getting almost full predation here, and that's pretty consistent. It doesn't say much for, obviously early season herbivory is what we care about in terms of getting our stand establishment and our yield. There's not much that a lot of these 3 pests can do in the fall that would negatively impact your yield at this point. There are other pests that you might get sporadically that will cause those yield losses. But what this is a good indicator of is that these predators were present and they were feeding like crazy, which, if to bring it back around to our slug activity density being lower in the fall than it was in the spring, this might be the reason why, that we're seeing this higher slug predation because we're keeping those predators in the system. And that's kind of the big game that we're trying to get to here is can we keep them in the system to make them consistently effective? And can we see this build up over time? So I'm right on time here as we go into kind of the wrap up. So just to kind of pull things together, this is really promising data. It's 1 year. We've still got a couple more years to go. These are small plots. I'd like to see this at some on-farm parts of on-farm projects moving forward. We have the paper that we just released is all on-farm data in Pennsylvania, and it's specifically focused on slugs, so no black cutworm, no true armyworm. But it shows kind of the same thing. So we're kind of seeing this consistently. We're seeing it play out in Pennsylvania on commercial farms. And so that does indicate that this might be a viable conservation ag practice across regions, and that it might help us manage pests in a way that we previously didn't know. And that's what it appears one of these major outcomes from this project could turn out to be. And again, it depends on what we see in the next 2 years. This is only insights from our first year. But if we continue to see this consistency, these are really good messages. They're really short, they're sweet, they're to the point, and they're convincing. To me, they're convincing. To the farmers we've spoken with in Pennsylvania who are conservation minded, they're convincing data. So we've got all of this moving forward, and that's really exciting. Just to reiterate, slug issues are going to continue to be problematic. In the mid-Atlantic, we're going to continue to get a lot of rain. We're going to continue to see slugs as a problem. They're going to expand in the Midwest as kind of flooding increases. John anecdotally has been receiving a lot more calls from the Midwest about slugs. And so that's another takeaway, is that this slug data doesn't just serve the Mid-Atlantic. This is really stepping out ahead of a problem that could really significantly impact field crop growers in the future. And so that's, again, one of these major awesome things about the PSA network is this is an anticipated problem. This is something that's a problem in one region that we anticipate getting worse. And our ability to kind of respond to that in real time and get an idea, a foundation of how problematic it could be is really important. And we'll be the first people to put out these type of data, and that's really exciting. And that's all I have. So really, what I want to do as we kind of head into the back quarter of this is open this up for questions, for discussion, for different types of analyses, for things to look out for in the future. And before we do that, I just want to thank the crew that helped me out this year in getting this all off the ground, getting it rolling at Penn State, and then the entire PSA team. This project is huge. There is a lot of moving parts. There are a lot of people managing different aspects of it. And we all, I think, based on these data, pulled off a really cool and awesome experiment. And whether, you know, it was a lot of work, but we're definitely going to be able to do this 2 more years in a row. And that's because of the people that we have in the CE2 team. So I want to send a huge shout out to all those folks who collected the data across the various regions and states we're working with.